So welcome back. So today we are going to discuss about site-directed mutagenesis or oligonucleotide-directed mutagenesis. Is a kind of application of recombinant DNA technology and a very important and interesting topic of protein engineering. Suppose in a particular individual there is a problem in its genomic structure, as a result of which he or she have some genetic disorder. Okay, and now by analyzing his or her gene, you find out that there is a change in his or her single base. Okay, we all know that our genome structure is nothing but a mixture of ATGC, right? So suppose in A, instead of having A, which is suppose a normal structure of the gene, he or she have AAT, and as a result the genetic disorder is occurring. Okay, so with the help of site directed mutagenesis or oligonucleotide directed mutagenesis, we can cure those kind of problems. How? We will simply remove that T and we will replace it with A. As a result, what will happen? Because we all know that our central dogma is telling us from DNA we will get RNA via a process called transcription. Then from RNA we will end up with protein via a process called translation, right? So what will be our genetic makeup of the DNA will facilitate the genetic makeup of mRNA, and what will be the genetic makeup of RNA will facilitate the makeup of protein, right? So if with the help of site directed mutagenesis we can change the makeup of the DNA, then it will reflect the change in its RNA, and also the change in RNA will reflect the change in protein. Okay, so that is how in curing genetical disease site directed mutagenesis is very interesting procedure. Okay, and not only that, we can also use this site directed mutagenesis or technique to see various kind of genes action in by creating transgenic enable. Okay, so that is the idea of site directed mutagenesis or oligonucleotide directed mutagenesis. So it is a interesting and important topic of protein engineering, where we are changing a single base. Okay, so it can be defined as a process that changes a single amino acid. Okay, and it is a kind of point mutation, we can say about this, because we are changing only a one base. So as a definition we can write that site directed mutagenesis, also known as oligonucleotide directed mutagenesis, is a process with the help of which we can change one amino acid of a protein or a single amino acid of a protein. Okay, so let's talk about what kind of things we required for performing site directed mutagenesis. First of all, we required a single stranded DNA molecule, that can be a plasmid, that can be a vector, anything that contain our gene that encodes your protein. Okay, that means here the T A T C G T T T C G T A G, this oligonucleotide codes for our protein. Okay, and if I zoom this picture, then this T will ultimately facilitate the synthesis of phenylalanine. Okay, because its complementary will be A. Although here we are doing a modification, I am going to talk about this. This A will facilitate the formation of UUU, and according to the genetic codon table, UUU signifies phenylalanine. Okay, So here our goal will be to change this phenylalanine into leucine. Okay, so for that purpose we need a single stranded DNA molecule that contains the gene of interest that codes for the protein. Okay, now on the basis of that gene of interest containing single stranded DNA, we will construct a primer, that is this one. Okay, now this primer will be complementary to the gene of interest encoding the protein, but here there is a little difference. Let me talk about it. So the difference is this primer will contain a base that will not complementary to the template. Okay, that means that is this one, it is not complementary to its T, right? So as a result, although there will be a mismatch, but it will not hamper the annealing because the other all bases are properly annealed. Okay, so that is the construct that we need. So now on the basis of that interesting primer, it will ensure the translation of a protein that will have leucine instead of phenylalanine in the protein. Okay, so let's discuss how it will happen. So here I have written that this is the oligonucleotide primer with one non-complementary base that will facilitate the amino acid change in the protein. Now here one of the bases is mismatched, but it does not causes any problem because other bases are well paired. Okay, so let's talk about how it will happen. So first of all, we will take a double stranded DNA molecule that will contain the gene that will encode the protein containing the normal amino acid. Okay, that is phenylalanine in this case. Here the AAA will facilitate the production of phenylalanine. Okay, so now by using denaturing agent we will make them single stranded. Okay, or by another means we will simply separate, make them single stranded. After that we will incorporate our primer with a non complementary base, only one non complementary base. Okay, so after that we will incorporate the DNA polymerase. So DNA polymerase will synthesize the another single stranded DNA molecule by extending the primer as we have here, but here still they are mismatched, right? There is no hydrogen bond between T and T, right? Then we will again separate the double stranded DNA molecule. Now we will have only the newly synthesized DNA strand. Okay, we will exclude the previous strand, right? Now we will, as another important thing I want to mention you, we have to know about the sequence of the oligonucleotide very carefully, right? Because after that our goal will be to synthesize a 3' end specific primer, right? Because we all know that DNA polymerase will do not perform the synthesis, but it can only perform the extension of the 3' OH. For that reason we have to provide a primer, right? So for that reason the DNA sequence information is importantly known to us, otherwise there is no utility of site directed mutagenesis. You cannot do site directed mutagenesis unless or until you know about your sequence very well. After that you will incorporate the 3' end specific primer, that is TAG here, and DNA polymerase will extend it. Okay, so as a result you will end up with this double stranded DNA molecule. And now let's try to focus this one, that is AAT. So AAT will produce a RNA that is UUA, and if you seen this one in your genetic codon table, then you will find out that it is codes for phenylalanine. Okay, sorry, it will codes for leucine that I have written here. Okay, so that's how you can change a single amino acid in a protein molecule with the help of the site directed mutagenesis technique. So again, I would like to recommend you to better understanding the site directed mutagenesis. Please learn about the genetic codon first. Okay, because this genetic codon will help you to learn about site directed mutagenesis. Then after that you can construct the site directed mutagenesis on your own. Okay, you can change any kind of amino acid, right? But for that reason you have to know about the genetic code. So if I will repeat this procedure, then site directed mutagenesis or oligonucleotide directed mutagenesis is a protein engineering technique in which we are replacing one amino acid from a protein, right? So here we are incorporating a point mutation at the DNA level. So what are the important thing that we have to keep in our mind? The one thing is we need a single stranded DNA template that contain the gene of interest that will encode the protein. Okay, and we have to know about the nucleotide sequence around the site to be altered. Okay, because if we know anything about what is at their surrounding, then we cannot design the primer, right? So we have to know about the sequence very well. Then we will require a oligonucleotide primer that will contain a one non-mismatched base. This non-mismatched base will facilitate the production of protein that will have the changed amino acid. Okay, now first thing is we need the double-stranded DNA molecule that have the gene of interest that encode the normal protein with normal amino acid. We will separate them. Okay. Now after separation, we will incorporate the primer along with the DNA polymerase. Okay. So DNA polymerase will synthesize another single-stranded DNA, resulting in the formation of the double-stranded DNA. Now this double-stranded DNA will again be separated and the older strand will be excluded. Only we have now the new DNA strand. After that, this new DNA strand we will incorporate the 3' in primer. Okay, and with the help of DNA polymerase we will extend in, and as a result we will end up with a double-stranded DNA molecule that contain the gene that will encode the protein obviously, and in the new protein you will have your desired amino acid instead of the previous amino acid. Okay, so that is the concept of site directed mutagenesis. So I hope this video will be helpful to you, and thank you for listening to this class.